Hello, it is an honor to be here. My name is Amanda Lin, and I am the leader of SCSHD. Our project is called AlphaGel. So just a little introduction on who we are. We are a group of biology enthusiasts from all over Suzhou and Shanghai. Our project started with a very cliche and romantic love story. Mr. Rambutan fell in love with a girl named Lychee. So she found out that she's actually allergic to Mr. Rambutan's hair. Sadly, they couldn't do anything about it until Mr. Rambutan said, hey, maybe I can do something about my hair using hair removal. And eventually he was successful and they fell in love and lived happily ever after. Okay, in all seriousness though, our project is dedicated to help find a solution to hair removal. Although we respect everybody's beauty choices, there is an increasing trend in demand for hair removal products. However, we found that sadly, some of the existing methods are very prehistoric and outdated. So we opt to find a cheaper and more convenient method to solve this issue. Just a little introduction about what keratin is. Keratin is a common linear protein found in the hair, hooves, wool, and other animals. And because of that, it is an increasing problem because there is so much keratin waste from animal products and agriculture. The problem with that is these wastes are being put into burying, landfilling, or incineration. And it proposes a lot of problems such as having a lot of greenhouse gas emissions and also public safety hazards. So the problem with that is we don't know what to do with it. And of course, planet Earth has provided a very smart, convenient method. We have microbes that have keratinase, which can degrade the keratin in hair. Hence the name AlphaGel, because we have alpha keratin in the vertebrates. Here are the problems with existing hair removal products. Hair removal cream, being the most common, leaves the skin irritated and burnt because it is made out of strong alkaline. Shavers, of course, are prehistoric, and sharp blades can often leave the skin scraped and has stubbles. Electrolysis, on the other hand, is expensive and extremely inconvenient, leaving the patient with no privacy at all. So we are looking for a better alternative. Here are some of the investigations we conducted in our earlier planning stage. Through research, we found that there is a surprising amount of confidence in our project. We found that people fully believe that it is very possible to create a project that is biologically hair removing. Hence, we found that people are actually interested in a hair removing product that is safe, efficient, and cheap. Therefore, we were very confident in our project overall.
Now, coming on to our experiment part, our objective is to create a hair-removing product based on degradation of keratin, and to achieve this, we do plenty of experiments from selection of suitable keratinase, inoculation, induction, purification, ultrafiltration, and final production of our packaging. To start with, we filtered our 4 proper keratinases out of all types we have researched based on their optimum working conditions, ensuring they're able to function efficiently under temperature and pH on human skin. Other than this, we also took induction time and conditions into account, so we could be doing experiments simultaneously to save time for repetition and modification. Now we choose 4 among all the enzymes so that we have a higher chance to finally synthesize an effective keratinase. With our newly manipulated plasmids containing 4 DNA sequences and 6 his tags, we specially added for purification purposes. We want to modify the E. coli BL21 to express these keratinases. Confirming the plasmids from the bacteria through sequencing, we then could manipulate induction followed by sonification and purification to check the expressed protein is our targeted keratinases. From researches and trials, we decided these values for induction. So we first applied the centrifugation method of a new resin for purification since the 6 his tags can be attracted by nickel. However, due to large loss of new resin, when removing the waste solutions, we changed to use NINTA columns. Here shows several tests we did so far, from a DNA sequencing test, So this SDS page that verifies we got the correct protein after purification, then to a series of concentration tests on enzyme. So after we have received enzymes produced and ultrafiltrated, we did groups of activity tests in order to find the most efficient way for keratin degradation. We wish to produce a fast and effective hair removal product, thus making sure our enzymes can be in the most active state is important. Since keratinases has shown activity on both polypeptide hydrolase and disulfide reductase, we could carry out tests on both protease activity and keratinase activity. To verify protease effect of all four keratinases, quantitative tests using three common natural protein substrates, casein, gelatin, and BSA, and one synthetic protein, azocasein, were carried out based on the following Seal-Cotters reagent and the method of spectral photometry respectively. After deplication, hair degradation effects of enzymes were observed and followed our experiments towards our packaging. So the aim of this series of protease tests was to select one or two keratinases out of the four that had a better effect with different concentrations of substrate. From this graph, we could clearly see that Ku3 and Q7 obtained a comparably higher degradation at high concentrations of substrate, and thus these two were chosen for further experiments. Now, moving on to activity tests, especially on keratinases, we mixed human hair with our enzymes. According to answers of questionnaires sent to potential customers during a human practice activity, we found it crucial to keep usage time of our products down to 30 minutes and focus to prove activity of our enzymes during this period of time. In this diagram, data supported the idea that activity would not decrease until 12 hours later, and up to this stage, we have proved that our enzyme can stay alive on customer skin. So our next step shall be to test if they could work properly in real conditions. Series of experiments observing human hair after keratinase treatment under microscope were done with control groups of reagent DTT and twist buffer. Promising results showed in this picture here that some gradient degradation effect appeared in groups of hair treated with both Ku3 and Q7. Hair samples were cracked instead of loosed, especially under the effect of a combination of enzymes. To better stimulate a human skin environment, porcine skin with hair was used as polymers present. Compared to control groups of DTT and twist buffer, surface of skin soaked in solutions of enzymes certainly changed, and hair on those pieces of skin was scrubbed off easily. This successfully proved feasibility of our designs. To ensure keratinases will remain active even after days of storage, suitable packaging must be chosen carefully. Based on suggestions provided by Coty Cosmetics Company, we took hydrogel into consideration. Activity tests would then help to prove its ability of keeping enzymes alive, and promising results appeared. In order to eliminate contamination, with the help of another iGem team, we decided to use smaller sub-packages, for example capsules containing hydrogel bead. As our final design of product, liquid hydrogel beads with an alginate coat was created. To test for feasibility of this design, we produced several capsules with Q3 and Q7 containing natural pigments of different color for attraction, and completed activity tests on enzymes in the center of beads. Both keratinases Q3 and Q7 maintained a comparably higher enzyme activity within the hydrogel capsule than common preservation in twist buffer. We did, including but not limited to, test for activity of keratinases right after they are produced. Enzymes were mixed with several possible packaging materials and enzymes with substrate of human skin hair. Furthermore, we plan to expand the diversity of our product, starting from analyzing efficiency in other media besides hydrogel. Our next step is to move into researches of cosmetics products to treat cornival layer, eco-friendly solution for zero dredging, and even into area of laser processing.
Now, I'll be talking about the different measurements that we took. First, we obtained results from experimenting with different concentrations of our enzyme, and we used the same artificial substrate, 0.1% acid casein, and waited 30 minutes before taking a measurement. To measure the solid concentration at the end of our reaction, we used our microplate reader to detect the OD value at an absorbance of 440 nanometers. To calculate our concentration gradients, we used enzyme concentrations of 0.51, 0.25, 0.125 in a ratio of 1, 1/2, 1/4. This will be more accurate than directly measuring with a certain concentration because we can reduce the impact of random and systematic errors. Then we performed an enzyme activity assay on our enzymes using the concentration gradient to compare and find the best 1 or 2 enzymes. In our next experiment, we controlled for the concentration of our enzyme and measured the different concentrations of our substrate to predict the optimum concentration for our enzyme activity. This was so that we'd understand how much enzyme should be present in our final product for quick and effective hair removal. To control for our results, we left an experiment with only acid casein, and we also tested Ku3 and Q7 on natural polymers such as gelatin and casein to examine the versatility of the enzymes. Through this, we found fairly promising results that indicated to us that enzymes were indeed applicable to more uses than just a singular function of hair removal. In the next section, I'll be talking about the different mathematical models that we use to better understand the properties of our keratinases. So the first model that we used was the Swiss model server, and we used this model to model the different structures of the 4 different keratinases that we experimented on. This was important because modeling the appearance helped us to understand their active sites and hydrophobic properties, both of which are important for the applications that we have in mind for our enzymes. The second model that we used was an ODE function that allowed us to model the translation, transcription, and degradation of our enzyme. And on this slide, you can see the different parameters that we put into our ODE function. And these were all measurements that we took in our lab. And here on this slide, you can see the way that each of these functions were derived using the values that we took. And the third, and one of our most important models, is our model of the Michaelis-Menten function. First of all, I would like to say that because our reaction uses a single substrate in free-flowing liquid over a short period of time, we were able to use the Michaelis-Menten equation because the underlying assumptions, such as the lack of back reaction and abundancy of substrate, don't really apply here. Because we know the concentrations of acid casein and keratinase, we can set up a time gradient to find Km, the concentration of a substrate at 1/2 Vmax, and this is also better known as the Michaelis-Menten constant. By using this function, we can understand the preferences of binding for an enzyme, and we can assess the biological impact that a particular enzyme has. For example, an enzyme with a low Km value will usually be found in an area with low concentration of its substrate, and that it will bind strongly. For the bottom graph, we graphed Ku3 and Q7 together on the same graph, while in the other graphs, we graphed the same enzyme on each graph. In fact, our final verdict was that Ku3 was the best enzyme. Our fourth and final model is our response surface analysis. By using this model, we can find the true impact of 2 very important variables, concentration and time, on the efficiency of our enzyme. So for one of our models, we found that we had an optimum time of around 2 days. And even though this isn't immediately useful for our cosmetic applications, we find that this might make leather processing and removing hair from animal skin more effective.
So I found that our human practice projects were very interesting because we took on the perspective of a beauty company and what they would do. So first of all, we did a lot of field research. We looked at future substitutes and competitors' projects, and we were looking at the products and their pros and cons, evaluating our own as well. Furthermore, we did research with consumers. We've tried to find out what they actually wanted. More importantly, we looked at a campaign and a marketing strategy that would help promote our project and gain more awareness. And lastly, and most importantly, we tried to integrate our human practice within our design cycle and improve our project. So the early stage of our human practice project was dedicated to developing a vague idea of what our project is and constructing some ideas. So we did a lot of things like the Beauty Expo Center in China, surveying some professionals in the area and what they would have done in our scenario. Also, we did things like the Plastic Era Charity Gallery, promoting our project and educating more about synthetic biology. The middle stage of our project is where we integrated our human practice within our design cycle. First of all, we went to a Coty Visit, which is a company that is professional in making makeup and skincare. We looked at the formula of our alpha gel and what we could have done to ensure our enzyme activity. The details are on our wiki. So we also did a cream review of competitive projects, for example, EGH, Veet, which are all very popular hair removal creams in China. We also participated in CCIC, which is the largest local iGem convention. We were able to reach out to hundreds of different other projects and teams all over the country. And because of that, we had an online meetup with teams that we were interested in afterwards. We were able to collaborate with NDNF and FAFU, which were all projects that helped develop our final alpha gel product. One of them helped us with hydrogel packaging, and the other helped us with the scenting problem with our enzymes because it smelt very bad. So the final stage of our project is refining it and making it a sellable product. So we had a meeting with Intercos, which is a very big makeup and cosmetic production brand, and we were able to better understand how a production line as well as a marketing project could be conducted. Afterwards, we used that knowledge and we did a speech on the product introduction towards a younger audience. Aside from that, we also had a lot of entrepreneurship projects looking like a beauty company. So first of all, we designed an exterior packaging for our product, which is shown here. I think it perfectly represents our colorful personality. We also did a couple of simulations of project shooting, which looked like this. Eventually, we had to put these information out in a product launch within our schools, and we were able to speak in front of people and get review. Lastly, we explored the possible implementation of keratinase. I think there is a great potential for this enzyme as a whole. First of all, cornification treatment, because our skin and hair is also made out of keratin. Furthermore, sewage treatment, because the biggest problem with especially girls is that we find that hair gets stuck in our drain. And lastly, and most importantly, leather treatment, because of many leather farms and industries wasting leather and not being able to treat it. And that is it. Here's our references. Thank you.